right? So within the module section of the 2018 paper, we're looking at it with the aid of a diagram, right, to explain how the information stored in a gene is used to synthesize RNA. All right, that is cool. What are we looking at? Which section or which I would say no. Which stage are we looking at? Transcription, right?
Transcription. Yeah.
Right? So looking at transcription specifically, hold on, give me a second, let me fix something real quick. Alright? So we're looking at transcription. Let me change something. Alright. So looking at transcription, what happens in transcription, guys? And so protein synthesis, right?
I think
Alright. So it's a protein synthesis. Go ahead, what happens? I'm looking for a diagram so I can place it there. So you can look at the diagram rather. Alright, so okay, I sent a diagram in the group last night. Let's just focus on what we're doing then. Right, instead of looking for the diagram. Alright.
So DNA helicase. Oh.
Alright, so what happens within transcription?
The DNA helicase on
So what
one.
happens within transcription? Wait, am I logging? Okay, so alright, hold on. Alright, so I'm not sure if I'm logging at this point. But what we're called to do is look at transcription, right? Explain how information in the gene is used to synthesize RNA, right? So what we're basically doing with the aid of the diagram, we're supposed to be looking at DNA strain, right? And what we're looking at is when we begin to look at the start codon or the primer, RNA polymerase would actually bind to the protein specifically with the help of helicase and topoisomerase, right? Which will break the bonds, the hydrogen bonds between the nucleotides, right? RNA polymerase will come in now and read each and every pair, well not pair, but each and every nucleotide on the DNA strand that it's reading, right, and produce a complementary RNA strand, right? And then after it's done now, when it reaches as end codon or a stop codon, it will come off and then the hydrogen bonds will reform. Go ahead.
Um, sir, so where does primase and ligase come in?
Oh, the kinase. What would kinase be? What's the role of kinase?
to join the sugar phosphate backbone.
Okay. What does kind mean? To basically bind, right? So what does kinase do, right? It really is kinase though. You know,
Sir, I thought you said like. Right?
oh, that's ligase. All right, well, kinase now. Where would kinase come in? Isn't kinase used probably within cell division more so than this?
No, sir, I said primase.
Oh, primate. Oh, all right. So I heard wrong. My apologies. So primase would probably put a primer, but primase usually has the advent of a primer. So we're gonna have like a DNA molecule, and then we'll have the a primer on it or a start codon, right? The thing is now within DNA replication, right, we'll see the advent of primase, but within synthesis now, right, where we usually see primase is that the helicase opens the strand, right, and topoisomerase, right, leads the helicase down the strand. primase is set on, right? Or we generally just have a start codon, right? Primase creates a primer, right, for the RNA polymerase to attach to, right? And then the RNA polymerase will now read up and create a complementary strand of RNA. And then when it reaches the stop codon, it will release. And then hydrogen bonds are reformed to form a double helix, right? So basically, we're supposed to create a diagram. What I would do here is just draw a DNA strand, right? And then in the middle, I draw an open DNA strand with a blob, I guess, an enzyme RNA polymerase on it with a different strand coming out. And then at the end now, I draw it basically closing back with the RNA polymerase leaving, right? So I basically just draw a step-to-step process of what is happening in my head, and then explain how that works, how DNA is important, how genetic information is stored. Because what the RNA is doing is reading creating complementary base pairs, right, from the DNA. So from there, you should be able to explain, right, that how the genetic information is stored within the gene to produce the RNA, right? So then think about the sequence. I know the sequence is a specific the sequence is really the information. The sequence has a specific code which the RNA will now provide to creation of proteins within protein synthesis, etc. Right? So basically, just describe how the information is used to synthesize RNA, right? So the RNA band that is produced will be directly in relation to the DNA nucleotide sequence. So you just need to speak about those stuff. And I think this question, oh my, 10 marks. Delicious. All right, so this question though, you need to work on your understanding of protein synthesis, right? I think this year, right? I'm not sure, but I think this year might have a lovely DNA replication question. Might. Or a lovely genetic engineering question. Well, let's look at this now. Using an example, briefly explain how proteins are responsible for the phenotype of organisms. Now, when we were answering this, right, we were playing right into the fact, right, of the central dogma of genetics, saying that DNA codes for RNA, which then codes for polypeptide chains, release amino acids, right? But here it speaks to how proteins are responsible for the phenotype, not how DNA is responsible for the phenotype, right? So proteins are the exact production of the genetic material of the cell, yes, right? But how do proteins refer to the phenotype? Proteins specifically the type of proteins produced will specifically speak to the type of enzymes produced and other types of structural proteins that will be produced for metabolic reactions as well as for general cell machinery. And if we change the types of proteins being produced, that will change how the organism itself presents, right? So based on the proteins being produced within the organism, that will change how the organism presents, right? So if the proteins are different, right? Let's say that the cell produces a different endoplasmic reticulum, right? Or it produces different cell membrane proteins because proteins are different, right? The structure of the proteins will determine how the organism goes through metabolism, how the organism uses cell communication, how the organism looks. So the different types and structures of proteins will be responsible for the phenotype of the organism. So we must be able to use those relationships without having to go back to DNA and how DNA and the genotype translates to the phenotype. Because through the proteins, so we must know how different protein structures can relate to how the cell presents or how the organism presents. So we need to describe that, right? Need to be able to describe that. That's just 5 marks though, so we need to be able to describe that. All right.
Sir, could you write out the answer?
Okay, the right of the answer. All right, let's see. Let's have a go at this answer. All right. So remember what we're truly looking at, right? All right. Okay. So what are we truly looking at, right? So we're looking at how proteins are responsible. Why is my brain lagging? It's responsible for a phenotype, right? So how are they responsible for phenotype? So let's look at let's take a bullet approach, right? So we know proteins. The proteins will determine, right? So you know structural proteins. Let's start there. Structural proteins, right? Proteins like collagen and tissues within the organs and stuff like that will influence physical traits. So different structural proteins will influence physical traits. Hopefully we can see that, right? Enzymes produce now different proteins will produce different enzymes, right? And that will know we know that enzymes catalyze different metabolic reactions, right? So it affects metabolism, right? And that in turn will influence phenotypic traits. Typic traits, right? So we know that as well. We could look at transport proteins specifically, right? So transport proteins, right? Could look at those as well, right? So the transport proteins like hemoglobin molecules and stuff like that. So the structure of your hemoglobin and different types of transfer proteins will influence the physiological health. Right? So the thing is no, right? I want you guys to understand how it works, right? I mean, it's exam time, so do you want to really understand or just pass the exam? But so just really looking at it, right? So the different types of proteins will lead into different characteristics, right? Even when you look at receptor proteins, especially for like cellular organisms, small cellular organisms, right? If you look at receptor proteins, right? How will that affect everything, right? They are the proteins that present on the cell membrane, right? So that will change how the organism looks, how it presents, right? It changes the regulations and how it receives signals from the cellular environment, and it influences the traits of immunity and sensory perception. Influence that. So basically talking about that, but generally what this syllabus wants us to look at is structural proteins, right? Because if my structural proteins are different from your structural proteins, I am going to look different from you. Oh, the mRNA carries the triplet codon as well. Yeah, you guys should look at that. Yeah, yes. Gordon, you should look at that, right? But yeah, so these are generally things, but generally I think the syllabus will be looking at specifically structural proteins. Because because a cat has different structural proteins than I do, right? So the cat will present differently than I do because the cat ultimately have different proteins, right? So we're looking at those type of things, how the different proteins will present to phenotypes of the organisms, alright? Outline the steps used in plant tissue culture and explain the key scientific principle underlying this technique. What are the steps? We have initiation, establishment, stuff like that. What happens in initiation, right? I don't know, I might just move through, right? So we have the selection, and we select suitable plant material, right? The writing is not, something to be gained, right? All right, so we're selecting suitable plant material, right? So usually it's from a meristem. Usually from a meristem, right? Some type of tissue that contains some type of growth, not growth factor, but stem cells, right? Looking at something like that, then establishment. Establishment. What happens in establishment now? What we do, we take those stuff, right? The tissue specifically, right? And we place it into a growth media.
Sir, won't you disinfect it? The explant
Oh yes, yes,
remove.
yes, yes, yes. Disinfection. Right, so we have disinfection. Sorry about that. You do have disinfection. Thank you so much. Right, so we're gonna have disinfection of the tissue as well to make sure that there are no viral components or any pathogenic components, right? We add to a growth media, right, with vitamins and hormones, right? And the hormones that we look at is
As-
auxins and cytokinins, right? Um, cytokinins, right? Auxins and cytokinins, right? What we do know, right? We allow it to grow. We have multiplication, or we could speak about just the growth, right? Into a callus. And what is the callus? Into a callus. And what is the callus? Undifferentiated cells, right? Go
Oh,
ahead.
yeah.
It is what? Go ahead, same way.
That it's a like a group of tissue or cell or something.
So it's a group or a ball of undifferentiated cells. Lovely. So it's a usually just a ball, right? It's just a bundle of undifferentiated cells, right? After that, now we have callus induction. I mean, it's really what we do for callus induction.
Is it that you're separating the callus to into individual cells or something like that?
So what we have is subcultures, right? So subcultures we're gonna have subcultures. One will have an increased concentration of what?
Cytokinin.
Okay, so let's start with cytokinins. Cytokinins, right? And that will promote what?
Root growth.
Root growth. Lovely. So we know the information, growth. And then the other subculture now will have an increased concentration of what?
Auxin.
Auxins, and it promotes what?
Shoot growth.
Shoot growth. For shoot growth. Lovely, right? And then we have acclimatization, right? What happens here?
That your is it the same thing as hardening? I think
Okay,
no.
okay, explain
Okay,
that. What is that?
I think it's when you literally take the I can't find the word
The
the
plant
tissue
extract, the tissue, okay.
out of the laboratory or the sterile environment that you're growing it and like introduce it to something like that.
Okay, so we take it out of that environment. We put it into a soilless media in a controlled environment. And this is usually in what? A greenhouse, right?
Yeah, green one.
Lovely. Usually in a greenhouse. So we know the information, we know the structures, we know the processes. Go ahead.
Is acclimatization the same as hardening?
Okay, what define hardening?
Sir, I think what I just said, that's hardening when you actually um take the plantlet out of the greenhouse or out of the area that it was being grown in and introduce it to the soil.
Well, okay, the thing is now with acclimatization, usually it's a soilless media, and then with hardening, we put it into soil after it grows and develops a little bit. Right? Because with hardening, what will happen after you have it in the controlled media, will adapt to external conditions before transplantation, or what we call as hardening. Right? So I usually call it transplantation, but if you know it as hardening, use the word. Right? You can have transplantation, or what you call hardening, where you take it out of that environment and put it into the field, into the soil. That's what you're proposing, right?
Yes, sir.
Okay. field. So soil. Lovely. So we have the process here. Alright, so we know for the this if it come, we know what's happening. And we know why with each of the things because I wrote down why we do each of the things. Lovely. Alright? 7 marks there. And then we move into the last component, the module 3 component of section B.